name is Mara. I'm a second-year PhD student in Paris at Sorbonne University and Institut Pasteur. And today I want to introduce to you my poster on a framework for identifying resistance-related gene expression signatures in single-cell RNA sequencing data of high-grade serous ovarian cancer patients. And my poster has the ID A209. So the project I'm working on is called the Paris project, which is a European project with some groups from Northern Europe involved. And the overall aims of the project are to identify biomarkers for high-grade serous ovarian cancer patient stratification and to identify precision drugs for overcoming chemotherapy resistance. Just to give you a bit of the context on high-grade serous ovarian cancer. So this is the most malignant and most frequent type of ovarian cancer. The 5-year survival rate is typically less than 50%. And the standard of care for the patients is debulking surgery combined with platinum-based chemotherapy. And what you can see here is the timeline of a typical high-grade serous ovarian cancer patient with the y-axis relative to the tumor volume. And you can see that initially patients typically respond to the treatment, but the tumor may eventually grow back, and over time the treatment becomes less and less efficient until finally the patients usually develop resistance. And how do we think about chemotherapy resistance? So there must be a certain subpopulation of cells within the highly heterogeneous tumor tissue. And this subpopulation, it survives the treatment and subsequently propagates. And these cells might be either already present before the therapy, which would be primary resistance, or these resistance may develop over time, over subsequent treatment, which is acquired resistance. But generally, there must be certain differences between the sensitive and resistant cells that make these resistant cells, basically resistant to the treatment. And we think that these differences are visible on a gene expression level. So this is a short summary of our work within the project. So our aims were to identify these chemo-resistance associated subpopulations of cells, and in a second step predict drugs that target the resistant subpopulations. And to give you a small hint on what our results are, we identified 2 candidate gene expression signatures that are highly associated with resistance, which I will describe in a second in more detail. But first, let me tell you how we got there. So here you can see our framework. Our assumption to identify the resistant subpopulations is that these subpopulations, they share common gene expression patterns. So what we do, starting with single-cell RNA sequencing data from multiple high-grade serous ovarian cancer patients. First of all, we identify general patterns of gene expression called candidate signatures. The signatures can be defined as just a set of genes with current expression as across a set of cells. Then in a second step, we filter for resistance signatures. Here we make use of multiple clinical variables and the fact that we have pairs of patients from before to after the chemotherapy. Then in the third step, we validate our candidate resistance signatures in external data. Here we make use of bulk RNA sequencing data from TCGA and a second external cohort. And then in a fourth step, we predict drugs that target specifically these resistance signatures. And if you want any more details, I'm very happy to discuss this with you at my poster, again, number or ID A209. So very briefly, the 2 signatures. This is the first signature, which could be that resulted from our framework when applied to high-grade serous ovarian cancer patient data. So this might be a potentially a copy number variation signature. We have here notably the 2 top genes that are located within the same genomic region. Why do we think it's associated with resistance? Because it is more highly expressed after the chemotherapy. And additionally, we see a negative association to PFI, which is platinum-free interval, a clinical indicator of resistance. And for the predicted drugs, the main mechanisms of action we identified are EGFR inhibitors. Then the second signature, we call it fibronectin-1 signature or extracellular matrix related signature. You can see that the main gene of the signature is fibronectin-1. Again, we think it's associated to resistance because it is higher expressed after the chemotherapy. And again, we see a negative correlation to platinum-free interval, this clinical indicator of resistance. And additionally, in the literature, we have found that fibronectin-1 has previously already been linked to resistance via the activation of notably the MAPK-ERK pathway. And also for the predicted drugs, we identified that the main mechanisms of action among the predicted drugs is the ERK-MAPK signaling pathway or MEK inhibitors. So to sum this up, we developed a framework to identify resistance signatures and to identify drugs that target these signatures. Then we've applied this to our single-cell RNA sequencing dataset of high-grade serous ovarian cancer patients, where we obtained 2 main signatures with a strong association with resistance. But generally, this framework is not restricted to high-grade serous ovarian cancer. It is generalizable and can be applied also to other cancer types to identify precision drugs for overcoming resistance. And as an outlook, so these results within the Paris project will go into the experimental validation in patient-derived organoids and PDX models.